All right, guys, Professor Davis here, and we are going to be looking at your genetics problems. This is Lab 8, and we're going to look a little bit at your assignment, but we also are going to do some practice problems to try to help you go through the process of doing these monohybrid crosses and dihybrid crosses. So if we take a look on your assignment, you'll notice that the first 2 pages give you lots of general information. So you can see that it tells you the instructions. You do get 2 attempts at this quiz. So you do get a chance to take the quiz, then you can go back and rework some of them and based on what your score is, and you can resubmit. You will also notice on here you have lots of review terms that are present. These are terms that are from the notes, and so you can see that you have gamete, progeny, allele, dominant recessive, homozygous, heterozygous. All these terms here are going to be important because they're going to help you be able to kind of decode what the question is wanting you to answer. You'll also notice as you go in here too, there's some general concepts that we're looking at. Okay, and there's also the whole idea of solving the problems. This gives you a step-by-step. And so this is a good reference tool on page 1 and on page 2 because it goes through how to do these problems. Okay, even gives you a YouTube link, that kind of thing, for the product rule of probability. Okay, so you've got those there. So I'm going to actually do a few practice problems with you first. We're going to be looking at just some genetic problems in general, and then we will set up a few on your actual assignment. And so these are right here, just practice. So let's kind of take a look. The big thing that I like to do when I do these problems is I really like to look at what it's asking. So in this first part, it says the following questions deal with a cross. And so I'm going to go through and highlight some important things here. So the first is the fact that it says homozygous. Okay, and so when we look at the idea of something being homozygous, remember that means they have the same letters. And it tells you that they're wired here with this capital S. So this means that I am going to end up having a dog that is homozygous, big S right here. Okay, and it's going to be crossed with a smooth-haired dog, which is little s. Now remember, if this is a recessive trait, it has to have the 2 little letters. So in this case, it's little s, little s. Now I'm going to exaggerate my little s's to make sure that you can see the difference. So this first part says, what is the genotype? Okay, so again, what's it asking for of the parents? That's these letters. So the first parent is big S, and the second parent is little s, little s. Okay, so these are our genotypes of our parent. Now, it's then asking what the genotype is of the offspring. Well, to get this answer right here, I have to do the Punnett square. All right, and so parent cross, this is big S, crossed with little s. And how I'm going to do to set up this Punnett square is I'm going to take these s's that you see right here, and I'm going to separate them. And I'm going to put 1 on one column and 1 on the other column. Okay, and then I'm going to take my next parent and I'm going to put them 1 of the little s's on each row. Okay, now when we do the Punnett square box, what you're going to do is you're going to end up bringing this s down and this s over. That's how the box is filled. And we always do capital letters first. So this is big S, little s. In this case, I'm going to bring this s down. I'm still going to bring that little s over. So this again is big S, little s. Okay. On this one, I bring this s down, my second little s over, big S down, second little s over. Okay, so now I have my actual Punnett square. Now, if we look at this, you'll notice every one of them is big S, little s. So this is 100% big S, little s. Okay, because all 4 boxes have that. And what does that mean? Okay, and so with the genotype, I already have the answer right here with this big S, little s. I can actually take that and place it in the box right here, or the line. But this is the genotype. If it was asking for the phenotype would be wire-haired. Now, they are wire-haired because they have that big S based on the instructions. Okay, and so that's what that means. If they have a capital S, they are wired-haired. All right, so this is just an example. All right, let's come down here to number 3. So in the F2 generation, what would be the phenotypic ratio of wire-haired to smooth-haired dogs? And so what we're going to be doing here is you're going to take 2 from the Punnett square above, and we're going to cross them. So big S, little s, because generation 2 means that we have to take them from generation 1. Okay. Now I'm going to set up my Punnett square. I'm going to have a big S and little s from parent 1 and big S, little s from parent 2. Right? So now I'm going to fill out my Punnett square. So this big S is going to come down. This big S is going to come over. This big S is going to come over. This little s is going to come down. Big S comes down. Little s comes over. And in our last little box over here, I have 2 little s's. Okay. Now, what is the question again asking for here? It's wanting what we would call the phenotypic ratio. So they want to know what they look like, not the actual genotype here. All right. So if they have at least a big S, they're going to be the wire-haired. And if they have the 2 little s's, they're smooth-haired. Okay, so since a big S is here, here, and here, that means that 3 of the 4, which is 75%, 3 of the 4 are going to be wire, and 25% is going to be smooth. Now, it wants what we call the ratio. So ratio means the base on the boxes. So 3 boxes are going to be wired, and only 1 is going to be smooth. So this is a 3-to-1 ratio, and that's what's going to go over here on the line, 3 to 1. All right, so this gives you just an idea of how to set that up. Okay. All right, so let's do one more monohybrid practice. Okay. We're going to look at this particular one. So this says grackles are robin-sized blackbirds which are commonly found in the United States. Suppose that 2 long-tailed grackles mate and produce a short-tailed grackle. Okay, so these guys were long-tailed and they produced a short tail. Okay. So what is the male's genotype? Now really it doesn't matter if we're talking about male or female. The fact that they were long and they produced something that was different than them tells me that the short tail is the recessive and the long was the dominant. So this means that they have to be heterozygous. They have to carry the recessive trait. So we're going to use the letter L here since it's long-tailed. And we're going to do, they have to have at least 1 big L, and they're going to have at least 1 little l. Okay, and the reason I figure this out is we have to look at the fact that if I set up this Punnett square, so we know they're long-tailed, okay, we may not know what this one is at this particular point because both parents were exactly the same, okay. We ended up with at least 1 little l. This tells us that these were the little l's. Okay, so if you figure out and do the whole Punnett square, the only chance is right there. Okay, so that's how the male genotype would be. So what is the probability that if these mated again, that they would end up having short-tailed? Guys, the whole thing has to do with every Punnett square has that same probability. So it's a 1 out of 4 chance. So it's a 1 out of 4 chance that they would have another offspring that had short-tailed feathers. Okay. All right, so we're going to move up. This will be the last one we do with the monohybrid for right now. This one says, a man can curl his tongue and is heterozygous for the trait. Okay, so again, we're going to break down the trait here. You see that they are heterozygous for the trait. And he marries a woman who cannot curl her tongue. This tells us that curling the tongue is dominant and not curling the tongue is recessive. Okay. So we're going to come down here and look at their Punnett square first. Okay. So he is heterozygous. Hetero means they are different. So he is going to be big T, little t. Okay. She cannot curl her tongue, which means she's the recessive little t, little t. All right. So I'm going to set up the Punnett square. Big T, 2 little t's. Okay, so I'm going to fill it in. All right, any questions on how we fill it in, please let me know. So now I'm going to come back up here and look at what they want. So what is the probability that they will have a child with their mother's phenotype? Cannot curl their tongue. Well, they have to have the homozygous recessive, and so there are 2 of the boxes. So what is the probability? They have a 1 out of 2 chance. Okay, half chance, 1 over 2. Okay, the next question though says, what is the probability they will have 2 children that cannot curl their tongue? Guys, this means they have, so 1 child and another. So here you're going to have to do 1/2 times 1/2. What's the chances? 1 times 1 equals 1. 2 times 2 equals 4. They have a 1/4 of a chance that both of their children cannot curl their tongue. Okay, so this right here is that law of probability. And there's a little video for you to watch on your worksheet if you are not understanding that. Okay, so this is the idea of a monohybrid cross. Okay, so now I'm going to take a little break of my practice and we're going to come over here and look at your actual 1 trait cross questions. So I'm going to help you set up this first part right here. So it says in a pea plant or in pea plants, a purple flower. Okay, so we know purple flower and it shows you right here, big P is dominant. And the white flower, little p, is recessive. Okay. What is the genotype of a pure breeding white plant? So guys, here when we look at the pure breeding, that means they are homozygous. So what does homozygous mean? It means they're the same. What 2 letters do they have to have if they're going to be a white flower? They are going to have these recessive letters. And so the white flower is going to be little p, little p. Okay. Now, if a pure breeding purple plant is crossed with the white plants, what phenotype is expected by the offspring? Okay, so I have my key here. Big P is purple, little p is white. The purple plant is still pure breeding, so that's homozygous. So that's going to be big P, big P. The white plant is going to be little p, little p. Now I'm going to set up the Punnett square. Each box is going to end up with a big P and a little p. So what phenotype is expected for the offspring? If they all have at least a capital letter, see the dominant is there, that means they're all going to be purple. Okay, so this is 100% purple. All right, do you guys see how we ended up getting that? You guys are going to do 3 through 5. I'm going to come down and do 6 and 7 with you. So let's come down here and look at 6 and 7. So in horses, we have 2 trotters. Okay, so we have 2 trotters. They're mated with each other and they produce only trotters. Okay, so they produced only trotters. 2 pacers are mated to each other and they produce only pacers. Okay. When one of these trotters is mated to one of the pacers, all the horses are trotters. Okay, so this is telling me that the trotters, because that was mated with a pacer and we saw trotters, that is the dominant. So big H here is going to be the trotter. Little h here is going to be the pacer. Okay, so trotters were dominant because that they were seen when they mated it with a pacer, and the pacers are going to be the recessive. So what is this expected phenotypic ratio of offspring that is produced when a hybrid mare? So guys, the hybrid part tells you it's different. Since it's different, we're going to have a big H, little h. Okay. And we also see that it's also going to be a hybrid stallion is mated. So he is also big H, little h. Okay. Now I'm going to take that and I'm going to set up my Punnett square. So I've got big H in the first box, big H, little h, big H, little h, and 2 little h's. So what is the phenotypic? So in this particular case, they wanted the phenotypic ratio. Let me look here. Of the offspring. All right. So since there are big H's in 3 of these boxes, 3 of them would be trotters. So you always put the dominant first. So I've got 3 trotters and I have 1 pacer. So the ratio would be 3 to 1. Okay. So this is how you're going to end up doing your... All right. So I am going to also help you look at... We're going to look at 12 and 13. Okay. So we're going to look at 12 and 13. So in pea plants, green pods, this is the big G, so green is dominant, and yellow, the little g, is recessive. Okay, so those are our keys there. Now it says when 2 plants with green pods are crossed, 25% of the offspring have yellow pods. What is the genotype of the green podded plants? Okay, so this goes back again. We're trying to figure out what these parents are. Now we knew they were green. So we have to have at least 1 big G in order for them to be green, correct? All right, and so if I put these in here where we've got at least a big G in the boxes that they need, okay? The issue here is it told you that 25%, which means one of the boxes had to be yellow. This is recessive. What do they have to have in order to be yellow? They have to have 2 little G's. There's only 1 box left that could have that. So this little g is going to come from up here, and this little g is going to come from over here. So the parents, this one was working backwards, the parents are going to be big G, little g. Both of them. Okay? So that's the first part. Now, for 13, it says if 220 plants are produced from the above cross, so this cross right here, how many offspring are expected to have green pods and how many are expected to have yellow pods? Guys, you have to take the percentage. There's 220 plants. So the green pods are going to be the 75%. So if you did 220 times 0.75, or you could do 220 divided by 3/4. You could use a calculator for that. That'll give you the number that needs to go here. As well with the yellow pods, you would do 220 times 0.25 because 25%. Make sure you do the decimal. Or you could do 220 divided by 1/4. Okay. That would end up giving you an idea, and those would be the numbers that go here. Okay. The last monohybrid one I'm going to help you with, we're going to look at number 17. What is the probability that a brown-eyed couple has 2 children with blue eyes? Okay, so let's talk about this right here. Brown is going to be dominant, so the capital B there is going to be brown. Okay, the lowercase b is going to be blue. Okay, so the thing is if some of these are going to end up having blue eyes, they could be carriers. So we're going to assume that they are going to be a heterozygous couple, okay? Meaning that they are going to have a big B, little b, crossed with a big B, little b. Okay? So now, if I go through here and I do big B, little b, big B, little b. This first one's going to be 2 big B's. Over here I have big B, little b, and 2 little b's. So guys, this first child that they have is going to have a 25% chance. Okay, so I've got a 1 out of 4 chance. Okay, the second child also has a 1 out of 4 chance because Punnett squares, remember, they don't remember what happened before. They don't keep track, and so because of this, we have to assume that the Punnett square starts over. So 1 out of 4. So this right here, guys, you're going to use the product rule of probability. Product means you have to multiply. So use that to help you get your answer. You have to multiply each of the chances of the kids. All right, so that helps you with your monohybrids. Okay. Now, the next little bit on this is going to be dihybrids. Now, before we get into the actual worksheet, I'm going to go back to one of my practice problems, and we are going to actually look at a dihybrid cross. Okay, so I have a dihybrid cross. In this particular case, it says about 70% of Americans perceive a bitter taste from the chemical phenolphthaleocarbamide, okay, or what they call PTC. All right, and so the bitter taste is going to end up being the dominant trait. So the dominant is that you taste it, and we're going to mark that with a big T. Not tasting it is going to be the recessive with the little t. Okay, and so we're going to look at tasting first there. But there's another trait we're going to look at, which is albinism. Albinism is also a single locus trait with normal pigment being dominant. So big A is the normal pigment. And lack of pigment is recessive with the little a. Okay, so I've got T's for taste, and I have A's for pigment. Okay, now I got to see who I'm testing. All right, a normally pigmented woman. Okay, so if she has normal pigment, we know that she at least has, so with the woman, we know she at least has a big A. Okay, who cannot taste PTC. She cannot taste this, so she's little t, because she cannot taste it, so she has to have the little letters. Has a father who is an albino taster. So the albino part from her dad tells you that he gave her a little a. See how the information, you have to break it down. Okay, so the genotype of the woman, we already answered 12, is little t, big A, little a. Okay, now she marries a homozygous, normally pigmented man. Okay, so the homozygous tells you that's the same letter and he's normally pigmented, so he's going to be big A, big A. Okay, he is a taster. Okay, so he is a taster right here, but he has a mother who does not taste. So if he's a taster, he at least has a big T, but since his mom cannot taste, that's the little t. Okay, so you have to break it down. It's like a word problem. You got to break it down. Okay, so this is the woman, this is the man. So then it says, what is the chance that their children will be albino and non-tasters? So when we look at this, they want to see how many of their children are going to be completely recessive. All the little letters. Okay, so we're going to cross mom. She's got little t, big A, little a. And dad, who's big T, big A, big A. And I'm going to show you how to set up this cross. Okay, so guys, we're going to do like what we would consider a FOIL in math. All right, and so I'm actually going to use a different colored pen to show you. Okay, so I'm going to take this first T and I'm going to pair it with that first A. Okay, and so that's going to be little t, big A. I'm also going to take that first T and I'm going to pair it with the little a. So I'm going to do little t, little a. Okay. Now that T is done. We've already used everything we can on it. So now I'm going to go to the next T and I'm going to pair it with the first A. So little t, big A. And I'm going to take it to that T to the next little a. And so that's little t, little a. Okay. So that's how I'm going to end up foiling it out so that I can set up my Punnett square. So let's look at the next one. I'm going to take big T to the first big A. So big T, first big A. I'm going to take the first big T over here to the second big A. Okay, now big T is done. So I'm going to go to the little t to the little a, or sorry, to the big A, and little t to the second big A. Okay, so that's how you set up the Punnett square. So now what I'm going to do with this is I'm going to put the letters together. T's go together, A's go together. Capital letters are always first. So in this first box, I'm going to put the big T, little t, big A, big A. Okay. The next box, I'm going to bring that big T over, little t down, big A over, little a down. Big T over, little t down, big A over, big A down. Big T over, little t down, big A over, little a down. Okay, do you guys see how I'm doing that? All right, so I'm going to do the next one. Big T over, little t down, big A, big A. All right, so I'm going to complete the rest of this. Now you'll notice since these two parents were the same here and all these were the same pretty much, okay, when we're looking at it, that's why these boxes look similar. Now this one's going to look a little different. I have two little t's, big A's, two little t's, big A, little a, two little t's, big A, and two little t's, big A, little a. Okay, over here again, two little t's, big A, two little t's, big A, little a, two little t's, big A, and two little t's, big A, little a. Okay. So now what we're looking for here to answer this question is we want one that's completely recessive, two little t's, two little a's. Based on these boxes, do you find any like that? Well, no, we don't. We have some that have the two little t's, right, here and here. But if you'll notice, there's never a box that has two little a's. So what's the chance that their child will be albino and a non-taster? There is a 0% chance. Okay, so this is how you set up a dihybrid. These are harder, so I'm only going to ask you to do 2 of them on your worksheet. Okay, so let's go back to the worksheet and let's look at the 2 traits. Okay, so we're right here. 2 trait gene problems. The following dihybrid crosses are going to be involved. And I'm going to help you with 18 through 21. You're going to have to, well, 18 through 20, you're going to have to set up 21 by yourself. All right, so if we come in here in this first set, in tomatoes, tall is dominant, short is recessive. Okay, so I'm going to come down here. Big T is tall, little t is short. Okay. Red fruit is dominant, so I'm going to put red with the big R. And yellow fruit is recessive, so I'm going to put yellow with the little r. Okay, so that's my key to start with, based on the information given here. All right, so then it says, what is the genotype of a tall plant? Okay, so tall plant that produces red fruit. And it tells you here that they are heterozygous for both traits. So remember, when you break down the word hetero, it means different. Okay, so we know it's tall, so we have to have a big T. Well, what's different from the big T? That's the little t. Okay, we know that it has red fruit, so that's a big R. And because it has to be different, then that's the little r. Okay. Now, if we look at 19, what are all the possible combination of gametes that could be produced by this plant? This is going to be where we do our foil technique, okay? So, big T can go with big R. So, that's my first one, right? Big T can go with little r. Now, big T's done. Little t is going to go with big R. And then little t goes with little r. So you have to have 4 options because that's how it goes across. Okay. Now, if 2 plants that are heterozygous for both traits are crossed, what's the expected phenotypic ratio? So I'm going to do 2 plants like what we just saw. So big T, big R, little r. Okay. Guys, you did the gametes right here on 19, so you're just going to take those and bring them down. So big T, big R, my first column. Big T, little r, second column. Little t, big R, third. And little t, little r, fourth. And the other parent is exactly the same, so I'm going to take those down the side. Big T, big R. Big T, little r. Okay, little t, big R. And little t, little r. All right. Now that I have them in here, I'm going to fill out my Punnett square. So bring big T over, big T down, big R over, big R down. This one had all capital. Over here I have two big T's. A little r is going to take the place of that second one. Big T, little t, big R, big R. Big T, little t, big R, little r. This one is going to be big T, big R, little r. Big T, little r, little r. Big T, big R, little r. Big T, little r, little r. Okay. Big T is going to come down because remember, capital letter always needs to be shown first. Okay. T, big R, little r. Little t, big R, big R. Little t, big R, little r. Big T, big R, little r. Big T, little r, little r. Two little t's, big R, little r. And all little letters. Okay. All right. So now what we're going to do is we're going to end up marking off the ones that we count because it wants the phenotypic ratio. So there are 4 options with this. Okay. We're going to look for tall plants that are red. We're going to look for tall plants that are yellow. We're going to look for short plants that are red. And then short plants that are yellow. Okay. Now, in order for the plant to be tall, we have to have at least one capital letter. And in order for it to be red, we have to have at least one capital R. Okay. So let's go through here. This first one has a capital T and a capital R. So that one is tall and red. So there's 1. Okay. This one also has at least one big T, one big R. So that is also red. This one is tall and red as well. Big T and a big R, tall and red. Big T, big R, tall and red. What about this one though? This one's tall, but what happened right here? The 2 little r's tells you it's yellow, so I can't count that one. What about this one with big T, big R? Yep, we're good there. This one, no. Big T, big R? Yep. Big T, big R? Yep. Nope, this one right here has 2 little t's, which means it's short. 2 little t's, it's short. Big T, big R. Nope, nope, nope. So I'm going to count how many I marked out. 1, 2, 3, 4, 5, 6, 7, 8, 9. So there were 9 of them that were tall and red. Okay. Now I'm going to do tall and yellow. Tall and yellow means I have to have at least one big T, but yellow tells you I need 2 little r's. Okay. Oh, here's one. Big T, little r's. There's 1. Big T, little r's. There's 2. Nope. Nope. Oh, here's another one. Big T, little r. There's 3. Okay, so I had 3 of them that were tall and yellow. All right, so red means I have to have a big letter with the R, but short means I have 2 little t's. Okay, so 2 little t's with a big R. There's 1, short and red. 2 little t's, big R. 2, short and red, and 2 little t's with a big R, 3. So there were 3 that were short and red, and last but not least, these are all recessive, so they're short and yellow, and there's only 1. So it says, what is the expected phenotypic ratio? This is a 9, 3, 3, 1 ratio. Okay, so this is how you end up doing the ratio. All right, so that's setting up a dihybrid cross. Okay, and you got one more dihybrid that you're going to do. Now the last thing that's in these set of kind of notes as you're going through to look at the types of Punnett squares is that you're going to look at X-linked, or what we would consider something that is sex-linked here. Now this one is a little different. It's very similar to a monohybrid, so just our basic cross. Okay, the issue here is because it is X-linked, you have to use the X's and the Y's. And when you give results, you've got to look at gender, male and female. Okay, so remember, in order to be female, XX is female, XY is male. Okay, so I'm going to set up this first one kind of here for you with setting it up, and then you are going to complete this Punnett square because this is a monohybrid. You should be able to do it. Okay, so in fruit flies, red eyes are dominant while white eyes are due to recessive gene. However, they are found on the X chromosome. So X big R is red eyes, X little r is white eyes. So what is the genotype of a red-eyed female that is a carrier of the recessive gene? Okay, so first of all, female means they are XX. So I'm going to put XX. She has red eyes, so she has to have at least one big R. Okay, the issue though is she is a carrier of the recessive trait, so she has one little r. That makes sense. Now for the male, it tells you here that the male is white-eyed. Okay, so males are XY. The Y doesn't get a trait here. But since he is white-eyed, he's going to be the little r. So X little r, Y. Okay, so that's how you figure that out. All right. So the next one. If a white-eyed female is mated with a red-eyed male, what percent of the offspring will be red-eyed? What percent of the offspring will be white-eyed? Okay, so I'm going to set up here. I'm going to help you put the Punnett square together, but you're going to have to come up with the answers. Okay, so for the female, remember this is XX. Now, since she has white eyes, this means she has the two little r's. Okay, now the male is XY. He's red-eyed, which means he has the big R. Okay, so now setting up the Punnett square, I'm going to put the female on top. I'm going to put the male down the side, and I want you to fill out the Punnett square. Now guys, on these questions, be careful. This first one asks you what percent of the offspring. That means it doesn't matter if it's a boy or girl, but how many of them are red-eyed? The second question says what percent of the male offspring. So you have to look at only the X and the Y's. Okay, so pay attention to that, and pay attention to that on 24 and 25 as well, because this whole page is X-linked traits. So make sure that you pay attention to that. Now guys, I know this video is a little long, but I wanted to show you some examples on how to set these up. And if you have watched this video, you also have part of your lab completed. There's no reason why you can't get those questions right on the quiz. So work on these, practice them. If you do have any questions over any of the problems, please ask me. Be specific though in your emails to me so that I can look at the problems and I can give you proper help. Again, if you have any questions or concerns, please let me know.